Hi all, this is going to be our third mini lecture for chapter 12. In this lecture we are going to talk about the neuroglial cells. So we've already talked about the types of neurons and the structure of neurons. Now we need to turn our focus to those supportive cells, which are our neuroglia. We'll look at what neuroglia we have in our central nervous system, as well as which ones we have in our peripheral nervous system. Talk a little bit more about, or start talking about myelin, which is going to be really important in nervous system function. And then in our next series of lectures, we'll move into how we actually carry out conduction of messages through a neuron and to its neighboring cells. So let's get started with our glial cells. Remember that in the nervous system, we have trillions of neurons. That's a lot of cells. However, those neurons are actually going to be outnumbered by neuroglial cells, or these glial cells, which are our supportive cells. We have a total of 6 types of neuroglia. 4 of them occur in the central nervous system and 2 of them occur in the peripheral nervous system. The term glia actually means glue. And these particular types of cells are often used for actually physically binding neurons together, as well as providing an overall structural framework. So they are sort of the glue of the nervous system, make sure everything functions properly. We're going to start by defining our central nervous system neuroglial cells. Again, there are 4 of them. So we have a couple of images here. This image in the upper right-hand corner, so over here, shows us a couple of neurons, and then it shows us all of the neuroglial cells that are associated with the central nervous system. This other one I'll talk about here in just a second. It pretty much just talks about one, or shows one of these different types of neuroglial cells. So let's start just in order that we have them listed on here. We'll start with our oligodendrocytes. So oligodendrocytes sort of have this bulbous body and up to 15 arms. They sort of look octopus-like sometimes. So here are going to be our oligodendrocytes. Here's that bulbous body, and then you can see we sort of have these legs that come off, or these arms. Each of these arms is going to wrap around a nerve fiber, so it's going to wrap around the axon of a neuron. And when they do that, they provide a myelin sheath. So I'm going to try and figure out a way to sort of write some of these details on here for you. Let's see. I might just have to do this one by one, but so we'll start with, We'll start with number 1, so that's going to be our oligodendrocytes. Again, these are going to wrap around nerve fibers. As they wrap around nerve fibers, they provide myelin. Myelin is an insulator, and it helps speed up conduction. Okay, and again, we are going to talk much more specifically about myelin in the next couple of slides, but I will use our laser pointer here to sort of show that. So here is the soma of a neuron. Here's that axon. You can see that we sort of have, they almost look like rolls of toilet paper, but they're these layers of myelin wrapped around the axon. Those are actually the arms of the oligodendrocytes. One oligodendrocyte is going to have arms that reach out to the axons of multiple neurons, and so you can see this bit of myelin here is actually from this oligodendrocyte. This next one comes from a different oligodendrocyte. So we have sort of multiple arms and multiple oligodendrocytes that are actually wrapping that myelin around that axon. So our next type of cell are going to be ependymal cells. Put that over here, and here are the ependymal cells. The ependymal cells they look kind of like a cuboidal epithelium. What they do is they actually line the internal cavities of the central nervous system, as well as produce cerebral spinal fluid and circulate it. So they are going to line Some of these things may seem a little bit foreign to us right now. We will specifically look at the brain and its structure next week. When I'm talking about cavities in the central nervous system, that will make a lot more sense once we've actually examined the anatomical structure of our brain. For now, when we are thinking about the brain in the next week, we can pull these two concepts together and think about the glial cells that are present lining those cavities, as well as producing and circulating our cerebrospinal fluid, which is the fluid that fills those cavities and helps keep the brain tissue healthy. Our third type of neuroglial cell in the central nervous system are called microglia. So microglia are small macrophages. They actually actively wander around the central nervous system and sort of probe around with all these tiny little extension-like arms and can remove debris and get rid of any cells that don't belong. So here is an example of our microglial cell. Again, these are, excuse me, small macrophages. And so these, again, are going to remove debris. So I'll sort of put that as down here. And the last type of neuroglia that we find in the central nervous system are our astrocytes. Here are the astrocytes. We also have the astrocytes shown down here. This is the astrocyte that we're talking about. So it sort of has this bulb-like or knob-like foot on the bottom of it. The astrocytes are going to be the most abundant type of neuroglial cell. They are sort of star-like in terms of their shape. So we have sort of a big body, and then we get lots of extensions or processes off of that body. It gives it a star-like appearance. They have quite a few functions. So they are going to give us a structural framework. They also have these feet, which is what I was showing you on here. So these feet are going to form what's called the blood-brain barrier. They help us regulate blood flow. They promote brain fluid composition and formation. They can fill spaces after neurons are damaged. So a neuron, if it is damaged, especially in the central nervous system, it cannot go through mitosis and it cannot produce a new cell or new neuron. If you have damage to a nerve cell or a neuron in the central nervous system, it is gone for good. We don't make new ones. We don't replace those. We don't fix them. They're simply out of luck, right? So they're out of commission. That's why traumatic brain injuries and spinal injuries are so problematic, is we don't replace the cells that have died. And so we don't just want open space though. So these astrocytes will come in and fill in with sort of the scar-like tissue. The one I want to go back to for the astrocytes is the blood-brain barrier. So in the brain, we do obviously get blood flow because we need oxygen there. Here is going to be a capillary that is serving the brain. Here is the squamous epithelial cells, endothelial cells of that blood vessel. Here's the brain. We also have these astrocytes and their little feet that are going to form a secondary barrier between the blood and the brain tissue. The reason that we do this is because sometimes small molecules from the blood can leak out of these capillaries and get into our tissues. That's usually not a huge problem for us, but in the brain is very, very sensitive to damage. If we have something leak out of the capillary in the brain and get into the brain tissue and damage some neuron, that neuron doesn't come back. So we have to be very careful. We only let very select few things. Again, these astrocyte knob-like feet are going to help prevent, so if something sneaks out between these two cells, it'll get bumped back into the blood rather than moving into the brain. This is a good feature for us. We like it for a lot of reasons. We don't like it for other reasons, and those reasons are typically pharmaceutical reasons. It is very difficult to find pharmaceutical drugs that can penetrate this blood-brain barrier and actually work on brain tissue. So it sort of poses a stumbling block for us when we're looking for drugs that we want to act on the nervous system. In the peripheral nervous system, we only have two types of neuroglial cells. We have satellite cells and we have Schwann cells. So again, here we have an axon for a neuron. Over here we have our two cells. So here's a satellite cell and here's the Schwann cell. The satellite cells are going to wrap around the soma within a ganglia. So a ganglia is going to be sort of a big mass of neurons that are only cell bodies. So the satellite cells wrap around those ganglia and they serve as insulation. They help regulate the chemical environment. So these are going to be and their job is going to be insulation. Oops. And the type of neuroglial cell in the peripheral nervous system that we're going to spend more time talking about are Schwann cells. Schwann cells are going to wrap around the axons of the neurons that are in the peripheral nervous system. They, similarly to what we see with our oligodendrocytes in the central nervous system, they wrap multiple times, so they sort of form this tight coil. So here we can see here is yellow is going to be the axon, and you can see that again we have this sort of tightly wrapped, almost like a roll of toilet paper all the way around that axon. That is going to be the Schwann cell. The Schwann cell is going to help us regenerate nerves, it's going to help insulate nerves, and it also produces our myelin. So myelin is present in both the central nervous system and the peripheral nervous system. The cells responsible for producing and maintaining that myelin are two different types of neuroglia. So I'm going to actually come back to this exact image in a few minutes, but I want to move on now and actually talk more specifically about what myelin is so that we can take a look at these two different types of cells in these two different locations and understand exactly what this means. Okay, so myelin is, like I've already said, sort of spiraled layers of insulation around the axons. It is actually composed of the plasma membranes of those glial cells, so either the oligodendrocytes or of the Schwann cells. Because they're made of plasma membrane, and if we think back to what plasma membrane is, it is going to be a combination of protein and a combination of lipids. So its overall composition is about 20% protein and about 80% lipid because it is the plasma membrane of those particular cells. When we have production of myelin, we call that myelination. Whoops. Yeah. Or we would say a cell is myelinated or unmyelinated. So myelin has a nice high lipid content. So in infants, most infants or all infants are born with limited myelin. I have this nice cute little video embedded on the slide that I can't show you, although you can watch it at home. It is my daughter when she was 14 days old, so exactly 2 weeks. It's her laying on a playmat, and it's her sort of moving around. And what I wanted you to see was how jerky and uncoordinated the movements of a newborn are. So they look like little robots. Their movements are very stiff and they just don't look quite right. That is because most of their neurons in their nervous system are unmyelinated. Myelin forms over time. When we have unmyelinated nerves, our conduction speed is slow. Whoops, did I spell real good? If we have myelinated neurons, we have fast signal speed. So if you've ever had slow internet and you're trying to load a video or watch something on YouTube, you know that it sort of loads partway and then stops, and then loads partway and then stops, and it takes a long time to actually send the entire message or watch that entire video all the way through. That would be slow signal speed. We started at one point, it takes a while to get to the end. If you have really fast, high-speed, good internet, that would be myelinated, where you can play something really quick, straight through. You don't have any pauses or breaks or slow speeds. So we do begin myelin production or myelination of our neurons in the fetus, but it's not completed until much older. Okay, so again, when you see a baby and you see how jerky, it's because their brain is sending a message to their arm to move, and it's just taking forever to get there. And then it gets there and they like move a little bit, and then they have to wait for the next message to get down that really, really slow pathway. So we're going to go back now and look at the myelin in the peripheral nervous system and compare that to myelin in the central nervous system. Here we are back in the peripheral nervous system, and we're going to pay most attention to sort of this bottom box here. This shows us mostly what's going on. In the peripheral nervous system, again, we have our Schwann cells. They spiral around one nerve fiber. So one but we do have multiple Schwann cells per nerve fiber. So this is one cell, this is one cell, this is one cell, this is one cell. This is all a single axon. But one Schwann cell is only going to be associated with one nerve fiber. They can wrap around up to 100 times, so we can get nice thick layers of myelin. There is very limited cytoplasm in these cells. Most of the cytoplasm is going to be in this sort of outer layer. So you can see in this cell how these inner layers are very thin. So they're very tightly packed together. And then we have a thicker layer on this outside edge. That's where all the cytoplasm and all of the organelles are going to be. Okay, so those are all in this outside layer. It's sort of like rolling a toothpaste tube up from the end, right? So you roll it up from the end and those first few coils are really thin, the walls are touching, and then all the toothpaste sort of accumulates in that outside layer. We call that region, it's not labeled on here, but we would call this region the that's an I, the neurolema. That neurolema is going to contain the nucleus and the cytoplasm and the other organelles. Okay, so let's compare this to our central nervous system myelin. So the myelin in the central nervous system is going to be a result of those oligodendrocytes. Keep in mind that we have a different ratio and structure here. We're going to have one oligodendrocyte and it has multiple legs that form the myelin sheaths around multiple axons. So we would have one oligodendrocyte to multiple axons or multiple nerve fibers. The myelination does actually spin in the opposite direction here. So it starts on the outside and spirals inward, as opposed to the central nervous system where it starts wrapped around the inside and spirals outward. In both the central nervous system and the peripheral nervous system, myelin is segmented, which means, I don't know if you've noticed this before, but we have a region of myelin and then a region with no myelin. Myelin, no myelin, myelin, no myelin, right? So in between each of these myelin sheaths, we are going to have a segment that is unmyelinated. Those segments, so this segment is called a node of Ranvier. And those nodes of Ranvier are going to be the gaps between our myelin. They are going to be very important in our conduction speed. So their presence is important for conduction. And I should also note that not all of our neurons are myelinated. When we talk about gray matter and white matter in the brain, white matter is myelinated neurons, gray matter is unmyelinated. So it really depends on whether we need speed in a particular area or not, as to whether or not we are going to use myelin. Okay, so I think you should be able to see this GIF that I have embedded over here. On the left is a neuron that is sending a signal. It is an unmyelinated neuron, so you can see the speed at which a signal moves down the axon of an unmyelinated neuron. On the right, we have a neuron that's the same size and diameter, and it is also sending a signal of what we will presume is the same strength, and you can see how much faster that signal travels down the myelinated axon. So on the right, we have myelinated. On the left, we have unmyelinated. So there are two major factors in how quickly a signal travels down a neuron. Those factors are how big the fiber is, so how big in diameter the axon actually is, and then the presence or absence of myelin. Our small unmyelinated fibers are going to carry messages slowest. Our large myelinated fibers are going to carry them fastest. And so here are sort of estimates of how quickly a signal can be sent down neurons of different size, as well as myelinated versus unmyelinated. So here would be two that are comparable, small, and then one's unmyelinated and one's myelinated. You can see 0.5 to 2 meters per second compared to 3 to 15 meters per second. So just the presence of myelin substantially increases the speed at which a signal travels down an axon. Okay, so I was going to spend a little bit more time talking about nerve regeneration, but this lecture is running long, and I think of all the things that we need to know about the nervous system, this one is less important than others, so I'm going to keep it fairly simple here and wrap this mini lecture up. So in the peripheral nervous system, we can regenerate nerves to some extent. That is only true if that particular neuron still has a cell body that's intact, as well as some of the neurolema of the Schwann cells, right? So we do have to have some remaining cell there, so the damage has to happen sort of low down in that neuron. This is a process that can take years, so up to 2 years to recover nerve sensation if a nerve has been damaged. And this, again, does not happen in the central nervous system. So I'm actually not going to go into the process here. You can sort of see it's outlined in these couple of images here, but I'm not going to expect you to remember the process of nerve generation, just sort of these points where we can regenerate, but it's iffy. If we have damage to the soma, not going to regenerate, it's just going to die. If too much of the cell is damaged, just going to die. It can be slow, and so, if you have ever cut a nerve, you may know that it can take 6 months, a year and a half to get all that feeling back as that nerve sort of slowly regenerates itself. Okay, so I'm going to end here. We're going to start in the next couple of lectures talking about our nerve signaling, and so those are going to be sort of the bulk of the rest of this chapter.